fungal phyla. I would say that 10 times fast. And their relationships with humans. So this is actually going to go fairly quickly since there's not a whole lot of detail that we have on several of these phyla. So it's fairly general for folks who are kind of wondering, oh my goodness, how do I study for this part of the course? Why do I need this? Well, all the information from the biodiversity section of the course, right? So that we just finished plants, we're in fungi, we're getting to animals next, is on every major professional exam you care to mention. It's on the GRE, it's on the DAT, it's on the MCAT, it's anything that you can care to mention for that. So that's sort of one of the reasons why you kind of need to know it. Studying for the exam, I highly recommend folks drawing out the phylogenetic trees and putting the characters on them. I know what folks have done is done one piece of paper per tree and then sort of connect them. So kind of like tape them together or something. So you could have the big eukaryote family tree, right? Just on the phyla that we, on the kingdoms that we were interested in, we zoomed in on, and then you can zoom in on plants, right, and then break it out and have one for plants and one for fungi and one for actually you're probably going to need more than one for animals. So just so you can kind of see how you should get essentially a bunch of nested clades within that. It's going to look like some really weird, funky looking tree when you're done. So as we saw with plants and their relatives, and we looked at protists, right, we have multicellularity is scattered throughout the eukaryote family tree, right? A lot of protists are unicellular, but then we have things like red algae and brown algae, right, which are multicellular. We have other examples as well. And the thinking is that this has evolved, pardon me, multiple times in our different eukaryote groups, so fungi, animals, others, and there seem to be multiple pathways to get to be multicellular. Another thing we also have to remember is that when plants colonized land, you're right, mycorrhizae, fungi were already there. So bacteria came first, right? The fungi followed next in sort of a classic primary succession pattern. And so, again, likely coming from an aquatic ancestor, and we have fossil spores here from fungi. Are the oldest ones that are definitive that yes, these are fungi ancestors are 460 million years old. So, several million years before plants arrived on land. The basal taxon is something called chytrid fungi, and we're going to get to them first here in our journey. They're the first to diverge, which is what, of course, makes them the basal taxon. And they also retain most of the ancestral flagellated protist ancestor characteristics. They actually have spores that still have flagella. Their spores actually swim. It's kind of creepy. And that actually has been a key feature for them in terms of being successful now. And I'll get into that here in just a second. What's also interesting about our chytrid fungi, most fungi have chitin in their cell walls. This was something that was very important in terms of retaining water as they came onto land. But a lot of our chytrids are still aquatic, and some of them actually don't have chitin in their cell walls at all. Okay, so within our kingdom fungi, we have 5 phyla that we need to be concerned with. They're still reorganizing some of these, but this is our current understanding of those taxonomic relationships. Both chytrids and zygomycetes may still be paraphyletic, so there may be some changes in the works, but we're going to go forward as is for the moment. So chytrids, of course, are our basal taxon, they are the first to diverge. So that's our oldest lineage. Zygomycetes are very likely the types of fungi you find on foodstuffs, so things like bread mold. We can see here a picture of mold growing on a strawberry. Other things on sort of vegetable crops are very likely to be in this group. Glomeromycetes, I didn't really mention at all, except for most mycorrhizae are in glomeromycetes. So that, of course, forming a very important mutual symbiotic relationship with plants. And then our last two taxa, ascomycetes and basidiomycetes, which together constitute most of the fungi that we eat, including things like morels and truffles, as well as sort of our regular run-of-the-mill mushrooms that you're likely to pick up in a local market. So I'm going to be going into details on each of these particular groups in terms of their features and how you can recognize them, and of course how they're different from each other. Okay, so who are our chytrids? These guys, as I mentioned, moist environments, they still have flagellated spores. Many of them are important decomposers, especially in aquatic ecosystems, but unfortunately a whole bunch of them are also parasites. And parasites of amphibians in particular. And so this is something that's creating a bit of a problem here. We have a nexus of some pretty nasty situations, and it's causing issues for a lot of our amphibian species are declining and in danger of extinction at the moment. So again, these are the flagellated spores, but they're also called zoospores. So there's something that's unique about chytrids. That's something they likely inherited from the ancestor and they retained, and the rest of our fungi have lost. Don't worry about the numbers and the letters on this. Actually, there are no numbers. The letters on this particular slide. This was just taken from a peer-reviewed publication studying the flagellated spores, and so I liked the nice close-up photograph, but didn't want to have to take the rest of it. So don't worry about the scientific name for this particular type of fungus, but I wanted to bring this up because you actually probably have heard about this in the news. Anyway, it's been a big deal. This is a chytrid fungus that is, again, it's you have the zoospores swimming, and they come across their host, which is an amphibian. Several different species of frogs are vulnerable to this particular type of fungus. It invades their skin, which is a nice moist surface, so it's a great place for the fungus to grow. The fungus also likes warmer conditions. And so this is with climate change, we're thinking this is why this is becoming an increasing problem. This is a fungus that's been around for ages. It's a very, very old lineage, but it's and our amphibians have been around for a very long time as well. So they have co-evolved together. This is not a new thing, but it's new in that it is increasing as an issue in terms of affecting population growth. And so individuals who are infected, often, again, you have a parasite, so you're having trouble getting food, you don't live as long, you're getting sick and dying. And what's interesting, again, we have two things. One, the climate change is making this worse. This is a fungus that was much more common in South America, and it seems to have naturally migrated up through Central America to North America, and is hammering our amphibians in North America that have not co-evolved with this fungus for nearly as long. And the other thing that's interesting is the mechanism by which it's doing this. So here we have another example of a parasite changing the behavior of the host in a way that's adaptive for the parasite, but maybe not so much for the host. And so the link here, for folks who are interested, go check it out. They have recordings of uninfected and infected males, and I'll be darned if I can hear the difference. But you guys with better ears probably can. In any event, the computer can, and the females certainly can. And so essentially what the fungal infection somehow is changing the call rate of these males, and basically changing their sexually selected vocalization to make it sexier to the females. And the females dig it, right? So then they come closer, and one thing leads to another, and now you have a way for the parasite to go from one host to another through physical contact. So it's a little bit, yeah, it's a little kind of funky situation. And unfortunately, again, those infected males don't live as long as healthy males. And so it's not good for the frogs, but it's certainly good for the parasite. Okay, so you'll notice that not a whole lot on this slide. Not a lot of detail about zygomycetes, other than these are the types of fungi that are most commonly found on foodstuffs. So fruits, vegetables, those kinds of things, most likely to be a zygomycete. Bread mold is included in that category. And I just love this photograph of here we've got right, these are the little fruits. So just so we remember, right, this is the achenes. I'm just going to draw and go. Okay, this is the strawberry fruit. Right, because remember, it's what we think of as the fruit is actually a swollen part of the stem. And then you've got the, obviously has been invaded by fungal spores that have then germinated and formed entirely new fungi, which have, again, grown. You have a mycelium under the surface, growing and absorbing nutrients from the decaying strawberry. And then, of course, when the conditions are good, they send up their own sporangia, right? So here's the fruiting body, is the stalk. Let me try to find a really nice one. Where's a good one? Where it's nice and clear what we're looking at. Okay, so yeah, here we go. So you've got the fruiting body is this guy. And so that's the fungal fruiting body. AKA, right, the sporangium. I'm going to say sporangia because there's probably more than one. This is the plural, right? Sporangium is singular. And so we have another, again, a mold here growing on an orange. So another example of a zygomycete. So the only reason I bring up glomeromycetes at all is because most of our mycorrhizae, right, which is the fungi that are forming that close symbiotic relationship with plant roots, so important to the colonization of land by early plants. Most of them fall into this particular phylum of fungi. So here we've got a photograph courtesy of the USDA Forest Service, who did some experiments with conifers, growing them either in soil that had mycorrhizae and soil that did not. It was sterilized soil that did not have any mycorrhizae. And you see, obviously, both plants germinated. They both grew. But the one that, right, obviously with the mycorrhizae, so this is the with mycorrhizae version here, much, much better, right? Was able to grow much larger and faster than the one that did not have that symbiotic relationship. So even though plants, can have true, most seed plants, I shouldn't say most seed plants, most plants have roots now. And they can absorb the nutrients and water all on their own. Having that extra help from the mycorrhizae is still super, super important for them to be successful. And so this is still an important symbiotic relationship. Okay, so ascomycetes, these are, you'll remember this name, right, when we talked about bacteria, and we talked about some types of, I believe it was gram-positive bacteria. We had two different types. We have streptomyces and we had ascomycetes. Myces, right, which is that suffix that's used with fungi because people thought they were fungus. So here are the actual fungi with the same name. So yes, it gets confusing. But clearly when you look at them, obviously you're not going to be fooled, right? If you look at, especially when you look at the fruiting bodies. So again, our ascomycetes are extremely important, decomposers, nutrient recyclers. There are some that are pathogenic, so that cause disease. And many, about 40% of ascomycetes are lichens. So most of the lichens we find in this particular phylum as well. And we know how important lichen are to succession. Right, so that's going to be actually the next slide. We're gonna look at lichen and be specific about them. So we call ascomycetes, right? They're named for the ascocarps. These are the little cup-like spore-producing structures. So we have the asci, so there's one there, right? And we have, more here. This is sort of a compound one. The one on the left here is a morel mushroom. This is something that is highly sought after for culinary enthusiasts. So it is an ascocarp, ascomycete, and not a regular mushroom as we think of. Most of those are basidiocarps or basidiomycetes. Okay, so here we have a little bit more about our ascomycetes and the lichens, right? So what a lichen is, right? This is actually a symbiotic relationship between two organisms. So it's actually not, we file them under fungus, but the fungus is providing the most of the tissue, but embedded in the tissue is either a green algae or a cyanobacteria that's living in the tissues. And so it's a symbiotic relationship. You have this ability to do both the decomposition and heterotrophic nutrient absorption of a fungus, and of course the photosynthetic capabilities of an green algae or a cyanobacteria, right? Because they of course have, cyanobacteria are the original chloroplasts, right? Green algae have them, so they can do photosynthesis. So again, our lichens are really, really important, both in terms of nutrient recycling and a nitrogen source. A lot of them do nitrogen fixation, right, because they have cyanobacteria, and those are champions at doing nitrogen fixation. But they also, through their decomposition, they also create soil. They break down granite, which is basically, pure rock. So here we've got a couple on the bottom here. This is an ascocarp. You can see the little cup-shaped asci there on the top. This one on the bottom right is a leaf-like lichen, so probably a cyanobacteria in that one. And then here we have an encrusting lichen, and yeah, this is growing on granite, which is volcanic rock. It's a metamorphic rock that's formed by extreme heat and pressure. So most often the kind of thing you find inside the heart of a volcano. So for example, Half Dome in Yosemite is what's called a pluton, and it's basically a giant granite mass, although interestingly it's made up of layers. It's not all one big piece. But there you have it. So all the volcanic nature of Yosemite. And you do see a lot of lichens breaking that rock down to form soil. Okay, which brings me to our last group of fungi, and I think these are the funnest guys. Ha ha ha. You guys know you miss my puns, right? You just know it. The groaning and the face palm and the oh, so bad. All right. So they are fun guys, though. They really are. Except, when you pick a bad one, right? So notice the guy at the top left there. You probably don't want to touch that, right? It's bright red. That just to me goes, I'm poisonous. And I honestly can't remember the name of that particular one, but it is toxic. So again, the differences we often pose between mushrooms and toadstools, even though they're both in basidiomycetes and they're both basidiocarps, is toadstools tend to be toxic, mushrooms tend not to be. But honestly, it's anybody's guess. So best advice is don't eat a mushroom or don't eat a fungus unless you're 100% sure that it's not toxic. Because the ones that are toxic can kill you sometimes even before you even know that you're sick with this toxic thing. It's, some of them are pretty nasty. I think there's one that's called Destroying Angel. So it's white. You have the death cap, right, which is another one that's also all white. So yeah, don't go be picking and eating wild fungus unless you're with a mycologist who knows what they're doing. And of course, a mycologist is someone who studies fungi, right? So again, our basidiomycetes are best at breaking down cellulose and lignans. So these are the ones you're most likely to find in forest conditions where they're breaking that down. Some of them are parasites, but most of them are just decomposers. And what's interesting, it's different a little bit than, again, we're doing producing spores from the fruiting bodies. Are doing this like the ascocarps and the fruiting bodies of zygomycetes are usually the result of, right, plasmogamy and heterogamy and all of that, the sexual stage. And you're producing the sporangium that's going to make spores by meiosis because the parent fungus is diploid and it wants to get back to that haploid stage. So most of our basidiocarps are the fruiting bodies that we eat. Most of the ones that you find are making spores by meiosis because they are the result of that sexual process of the plasmogamy and then the karyogamy. And what's also interesting, a defining feature of basidiocarps, separate from ascocarps, right? There's this sort of a cup-shaped sporangium. In basidiocarps, you have these little gills underneath, and that's actually what they're called. They're called gills. I know, go figure. But they're thin, and as they're this thin mesh network, which is very, very similar to the structure of actual fish gills. So it sort of makes sense. And that entire surface is, all those cells on that surface are that we see on the underside of this mushroom here on the left, are going through meiosis and making spores. And I actually have a close-up of the cells that are doing that here on the next slide. So I just grabbed this photo off the internet, but if I'm not mistaken, I think this is a death cap on the left. And what it's doing is going through meiosis, right? So we have the basidium, basically the fungal version of a sporocyte in a plant going through meiosis. And so, of course, you're getting 4 spores as a consequence of that, right? Because you've gone through 2 rounds of cell division, and so you get 4 haploid spores that are genetically distinct from each other and unique. So good chance for additional genetic recombination there. So most of our fruiting bodies in fungi, and you really would have to know, you look at zygomycetes, ascomycetes, and basidiomycetes, most of those fruiting bodies are the result of plasmogamy and followed by karyogamy and then making a sporangium. that's going to make spores through meiosis, haploid spores. In other types of fungus, though, the fruiting bodies may be, the sporangia may be a result of just the conditions are good, and you have a haploid fungus, and so that haploid fungus is making spores through mitosis because it's already haploid. Okay, so I mentioned that several of the fungi that we were looking at had either were either parasites or pathogenic species were in this particular group. And of course, a pathogen is anything that causes disease, right? So this could be, you know, a virus, it could be a bacteria, or it could be a fungus, or other kind of animal parasite, right? Or any kind of protist, right? We talked about several different kinds of protists that are adopted a parasitic lifestyle. And cause disease like malaria, you know, or Chagas disease, for example. So a fungal infection is just broadly termed mycosis. Again, a mycologist is somebody who studies fungi. So this includes, you know, again, fungal infections that we commonly get. Our bodies are covered in you know, little spores, right, all the time. So there's athlete's foot, right? Yeast infection. So remember, we have single-celled fungi, right, and that reproduce asexually, and we call them yeast. They're still eukaryotes. They're still fungus. They're just single-celled. There's a very nasty fungal agent that causes pneumonia, which makes it rather interesting, right? Because you actually have three different forms. You have viral pneumonia, you have bacterial pneumonia, which is a pneumococcus, right? And you have fungal pneumonia. And these symptoms are extremely similar between all three of those. And so it helps explain why it might take a long time to diagnose somebody. And then you add on, of course, the novel coronavirus on top of that. And it makes it difficult to figure out exactly what is causing someone's symptoms. And of course, not everybody gets exactly the same symptoms, just to keep it interesting. So my heart goes out to the medical professionals trying to deal with all of this stuff and figure out what to prescribe someone, right? Because you know, is an antibiotic going to help someone who has a fungal infection? Yeah, and you're like, okay, fungus among us. Okay. Sorry. So again, if you're keeping track of the bingo, I've just laughed at my own joke. So there we go. But again, it's not all bad. So again, we have fungi that cause disease, but you know, fungi again are great sources of antibiotics. Historically, extremely important. There have also been really good sources of immune suppressant drugs for organ transplants. So folks who are trying to keep your immune system from attacking and rejecting your new heart or your new kidney, right? Yeast we use in brewing and baking the world over. Anything that's leavened, right? We have unleavened bread. That means there's no yeast. Leavened bread is typically what we're using, and you have bread that's risen. It's gotten all poofy. You have little holes, like we see the Swiss cheese here in the bottom right. Those are holes that are from yeast doing fermentation, and as a byproduct of fermentation, they produce carbon dioxide, and carbon dioxide bubbles form in the cheese, and so that's where those holes are coming from. So really important for that purpose. And just as an interesting interesting aside, so in addition to, you know, several different kinds of medications, we also get some other interesting drug components come from a fungus. So a particular ascomycete that grows on rye gives us the components of LSD or acid. Okay, so as promised, the link between fungi and animals, reinforcing that. So we've already been here, right? So again, most of our protists are sharing that, that share a close common ancestor with animals. And fungi also have flagella, so it's not a wild stretch to hypothesize that we are coming from an unicellular flagellate protist ancestor. Remembering a protist is just a eukaryote that's not a plant, an animal, or a fungus. So this is, of course, before you had some divergence, and we have two different lineages that take two different trajectories. One goes towards fungi and nuclearids, which, remembering, those are amoebas and things like that. And then the other branch going towards animals and choanoflagellates. And of course, reinforced with the DNA evidence that does seem to suggest that we are dealing with a nice monophyletic group. So unlike some of our other taxonomic sequences where it's a hot mess in terms of figuring out who's more closely related to whom, we do appear to have a nice monophyletic group in this instance. Okay, so what the heck is a choanoflagellate, right? Well, as you say flagellate, you're probably thinking, well, it's something that's got a flagella. I mean, I know that much, right? And that's, of course, that long squiggly bit, right, in the photo, in the drawing here. These are largely single-celled flagellated protists. But what's interesting is we have another possible pathway to multicellularity. Because as you see, right, they're now coming together in a little group there on the right-hand side, in little clusters. And so we're thinking these living in groups, and then they start cooperating, right? And so we have another pathway to multicellularity there. The DNA does also back up this close evolutionary relationship between choanoflagellates and animals. But in all honesty, the reason I'm even talking about them at all, and I'm not just skipping them, is the close tie that they appear to have with our basal animal taxon, which is sponges. So when you look at these little choanocytes, right, the collar cells, we're looking on the bottom right. These are the choanoflagellate collar cells. Next time when we look at the choanocytes of sponges and you put them side by side, they're almost identical. They're so similar. It's not even funny. So it really does suggest either we've got, again, a complex characteristic that evolved in identical ways in two different groups, which your BS meter is probably going off, going, yeah, that doesn't sound terribly likely, right? I mean, yeah, even if it does happen, there tends to be little differences that pop up that give you information that it's coming from two different sources, right? They're made of different proteins, right? Or they're constructed in a slightly different way, even though they do the same function. No, we have things that are almost completely identical between these two groups. And so that really does suggest a strong, close family relationship here between choanoflagellates and animals, given that sponges are our basal animal taxon and have a lot more features in common with actually choanoflagellates than they do with a lot of other animals, or a lot of animals, actually, to be honest. What's also interesting is that we see cells that are similar to the collar cells in animals, but not in fungi or plants, right? Or other kinds of protists. So that also suggests a close family relationship. And a recent discovery, I want to say this is about 3 years ago, folks who were looking at the DNA sequences between choanoflagellates and animals were finding genes that were thought to be unique to animals, and they found them in choanoflagellates also. So, so we have what appear to be homologous gene sequences involved as well. So the DNA backs it up, right? The structure of the collar cells back it up. And the fact that we have, again, proteins and things that are only found in these taxa and not elsewhere is indicative of a very close relationship.